Great. So thank you very much for this opportunity. And I'm humbled and honored and a little bit intimidated to be giving it to all of you who are experts in neurology and immunology and medical genetics. But I feel pressure if there are AT families who will be listening to this, because I know that for them, time is very short and they're very eager to hear of progress. So, but again, I want to make it clear, I'm not a physician or a scientist. I went to Harvard Business School, but I went into a business. My first business was the food industry. My main product was shrimp. So that has nothing to do with molecular biology or genetics, and I didn't know how medical research was done. I thought I had a pretty good life, maybe a perfect life. One point in this picture, I had a nice wife and 3 beautiful sons. But like many AT families, then we were surprised with a diagnosis. And so the 2 boys on the right, Jarrett and Quinn, were diagnosed with AT. As all of you know already, it's a very rare, obscure disease. We call it an orphan disease. No one famous or wealthy or powerful has ever had a child with AT. Most people in the United States, even doctors, physicians, don't know too much about AT. And as you know, it affects many systems in the body. Today, I'm not going to talk about what we know about the phenotype of AT, immunology or neurology too much, because all of you are familiar with it, and you can certainly read review papers to get the details. But what I want to do today is just tell you my story and the story of the AT Children's Project. I think it's good for families and family organizations in different countries to compare and learn from each other's efforts, and maybe that can be helpful today. At the same time, I'll explain a little bit about the research that we are supporting right now. So as you know, AT children experience cerebral atrophy that causes often a misdiagnosis when they're very young, because it can appear static, like it doesn't change, but eventually the deterioration causes loss of muscle control and wheelchairs, swallowing and eye tracking, all these things. I think the important point is that even though on the right you can see the child on the bottom has AT, 12 years old, the bottom on the top right doesn't. We now know that AT probably affects many more areas of the brain. Clearly, the brainstem and basal ganglia are affected. When you look at the cells that are dying, in the past we focused on cerebellar Purkinje cells or basket cells that were dying. Today we know also a lot of glial cells die. So my response after my children were diagnosed was to learn the science some. I had 2 PhDs who were very kind and taught me a lot of science, not so that I could do scientific research, I could never do that, but so that I would not waste people's time. And so I wanted to learn the science that was relevant to AT at that moment. So when we first began the AT Children's Project, I started to learn about molecular genetics. Back then, it was linkage studies and positional cloning. After the gene was found, I didn't care too much about genetics. I moved on to making animal models or raising antibodies to localize the protein and so on. So I've been trying to learn the science, but even more important, I've been trying to learn how the science works. Lay people like me that are not in the scientific world have no idea how scientists are funded, where they get their money, and how research is supported by governments or by companies. I had to learn that. I also had to learn who the scientists were. So there were about 4 or 5 groups that were looking for the AT gene. There were many other researchers who were interested in DNA damage. And in the early days, AT was most well studied by radiobiologists who were interested in radiosensitivity of AT cells. But I had to learn who the scientists were, and somehow I had to understand maybe which scientists were more rigorous or more making more progress. If you ask them, they all say they are, but it was difficult to figure that out. I also needed to learn how scientists are motivated. All people are different. So some scientists maybe want to be famous. Some scientists are very compassionate and want to help children they meet with AT. And some scientists, I think many scientists, people who go into the field of science and research, are motivated by solving a puzzle. They're very curious people. So I needed to learn what would make scientists want to work on AT. And then learning how to fund the AT research was the biggest challenge for me. I do not like to raise money from donations. I'm a businessman. Asking for gifts is very difficult for me, but we had to learn that. And so we started an organization called the AT Children's Project. We called it the project instead of the foundation because we wanted to use a more temporary word. We were hoping that this organization would only be temporary and find a treatment and then stop. I have always been a volunteer, never paid by the AT Children's Project. I'm just a father, and I have in the day I work with companies, but in my night times I'm a volunteer, and I've been involved with the AT Children's Project. So we began having many different strategies for raising money, and I can talk some other time about the different fundraising events and ways that we increased awareness. But many families around the country began to help. With a really rare disease like AT, we learned that you cannot depend on only families who have a personal connection to AT, maybe a child with AT. Instead, we really had to reach out to people who have healthy children and maybe have them help us. They maybe because they are grateful for having healthy children, but they can understand what we are going through, they helped us. So a lot of kind people have helped us raise funds for AT. We also increased awareness many different ways with newspapers and magazines and now social media. You can see on the bottom, we even had one man who took a sailboat across the Atlantic Ocean by himself to increase awareness for AT. And then with the funds that we raised, we began supporting AT research. So we've never cared about where the researchers were. I don't care about being an American organization. If there was a researcher like Yossi Shilo in Israel who found the gene, or researchers, we have funded in the past researchers in Turkey. We've also funded them in Asia, many different places. And all we care about is novel approaches, rigorous scientists, and really ideas that could have next steps if they're successful. An important role that we played was to collect and share biospecimens like blood samples, skin fibroblast samples. We found in the early days that sometimes researchers created these banks of tissue samples, but they would not share, or they would require to be an author on the paper, and a lot of negotiation. And I thought that was ridiculous. So we set up banks, public banks, where these cell lines that were created could be shared immediately to any researcher in the world who became interested in AT. We also began to support making different kinds of tools for research. So over the years, that has been different things, but early it was antibodies against the ATM protein. When iPS cells became more popular for research, making induced pluripotent stem cells, and then differentiate them into different other cell types. We found it was important to make iPS cells available from AT kids. Those are some examples. And then we use these tools and tissue samples to really enable access to all researchers. Again, we did not want access to samples to stop people from being able to work on research. If any researcher had an idea, for example, maybe a very respected DNA damage repair scientist became interested in AT, we wanted it to be very easy for him. Over the years, we've supported a lot of different efforts to create animal models of AT, knocking out the ATM gene. In general, this has been very frustrating. We've made them in all different types of species, from yeast and fruit flies, Drosophila, zebrafish, to even making pigs. In many different strategies, more recently with CRISPR, which is easier to make knockouts. But the AT mouse has recapitulated or displayed the the immunological problems and a lot of the cellular phenotypes, radiosensitivity, and the cancer risk. But frankly, the neurological deterioration we see in children is not seen in AT mice. We thought it might be strain differences, so we funded researchers to make knockout mice in 15 or more different strains, background strains of AT mice, but that did not seem to make a difference. Sometimes a researcher publishes a paper saying that it has a neurological phenotype, but it's very, very subtle. For example, they put the mice on a rotorod, and the wild type mice, fall off in 70 seconds, and the AT mice fall off in 68 seconds, and they say, oh, it's a neurological phenotype, but it's not a good model for AT. The good news is very recently, in the last 2 or 3 years, we have had 2 research groups make AT mice by knocking out a second gene that's in a related pathway. And by doing so, both knockouts separately do not have a phenotype, but together they have a clear neurological phenotype. And we think that this mouse model that is now at UCLA in California, but also at St. Jude Children's Hospital in Memphis, Tennessee, those 2 labs, the mice that they've created, we think are pretty good models for testing therapeutics or for understanding the mechanisms of AT. So additional things that the AT Children's Project has done is we've organized a lot of meetings. So sometimes very large meetings with poster sessions and abstract books, and sometimes very small focused workshops, focused meetings on one subject. For example, last November, we had a meeting that was a smaller meeting with about 40 people in Boston, focused on gene therapy for AT. We created a data platform for AT so that we could collect a lot of data in one place. Again, it's not an American database, it's an international database. The families that oversee it I'm the only American parent. 12 other families are all from other countries. And the purpose is to really bring together a lot of data. We also created clinical centers. So somebody mentioned Dr. Crawford coming to Turkey recently. When we started the AT Children's Project, typically one doctor would only see maybe one patient in his or her career, So they did not have a good natural history. They did not have a good idea of what was typical for AT. They did not know about milder variant cases of AT. So it was important to have a clinical center that was multidisciplinary with a neurologist, an immunologist, a pulmonologist for the lung problems, maybe a swallowing expert, speech expert, and in an institution where you could have other collaborators for other things like eyes. Oncologist is also important. So I want to mention the global AT family data platform that we have. So, we've now got over 629 AT patients who their families have participated in surveys and completed data information. In some cases, they've added additional records and so forth. We've also done whole genome sequencing from some countries. Many countries are willing to do this. Some countries, I think Turkey, it's very difficult to have any DNA or DNA sequence data leave the country. But for those countries where we can, today we have about 390 whole genome sequences. And this is useful, of course, to look at the ATM gene to see what kind of mutations patients have. But as I will discuss later, it's also useful to look for modifier genes across the whole genome that may be associated with differences in phenotypes. If we do identify a modifier gene, it could then be a potential therapeutic target. So there's just the data platform we have, atfamilies.org, has now been translated into 11 different languages, but we continue to expand that. So we've done a lot of clinical studies to look at lung function in AT, swallowing, eye tracking. Are there exercises you can do for swallowing that would be good, for example? And even looking at cognition, neuropsychiatry, neuropsychology. We've done brain imaging studies. Of course, most AT patients have had MRI scans done. For many years, everyone told me that we could never do PET scans to look at, for example, FDG PET scanning to look at metabolic activity in AT children's brains because everyone told me that PET imaging, the radio label would be toxic or dangerous for AT children. Eventually, we spoke to some experts in PET imaging and calculated the exposure of radiation and determined that it was safe for AT families. So we have done PET imaging now on AT patients. Right now there is an effort underway to not just do activity, like FDG, but also, for example, a study of TSPO, which is a PET tracer for possibly for neuroinflammation. And we'll talk later about a PET tracer we're developing against the ATM protein so that we can determine if gene therapy is working. We began looking for biomarkers. Clearly, with a disease like AT, where you have slow progression, to run a clinical trial of any kind, it's really ideal to have a fluid biomarker you could follow, a surrogate marker. And then we also have held caregiver workshops, so meetings where families and people who take care of AT children can come and compare notes, discuss strategies for making life better for AT at all ages. We've also created handbooks that we share that AT families use who are new to AT and have to understand about school or about vaccinations or about how to care for different aspects of AT. But also these books are useful for sharing with their teachers or doctors. Today, we are continuing to look for biomarkers for AT. So a couple of years ago, we assayed neurofilament light chain and serum of AT kids and found that NFL is dramatically increased in AT children. We are now expanding that study to a wider range of ages and also longitudinally to see if it changes over time. One could expect that maybe NFL goes up during the time of greatest cell death in the brain, since NFL is a marker for cell death and axon damage. Maybe the NFL levels will be the highest between ages of 6 and 12, and then maybe plateau. We don't know. We'll learn this soon. We are also beginning to check other well-known biomarkers for neurodegeneration. So you can see some of the listed here with phosphorylated tau and other kinds of neurofilament light chain. But our hope is that we can find one or two that are maybe useful and truly are surrogates for clinical readout. In the United States, when the AT Children's Project was started, most of the time, the typical age of diagnosis was 3 years or 4 years old. This is, if a child was a little bit wobbly, had a little bit of a problem, it was frequently misdiagnosed as cerebral palsy or static insult to the brain. Unless the family had a second child with AT, then maybe they looked at genetics. That's what happened with my family. But more recently in the United States, and it is now common to do newborn screening for primary immune deficiency. And so in the course of doing that, a lot of babies with AT are caught in the screen. And then when it's clear that they don't have SCID, they don't have immune deficiency, primary immune deficiency, there's an algorithm and they end up being genetically tested and confirmed to have AT. So the age of diagnosis for AT in the United States has dropped significantly. We now are identifying children who are 2 months old or 3 months old instead of 4 years old. This is, for me, a good thing and a bad thing. So the good thing is that if we have some kind of intervention, like a gene therapy, most of us believe that intervening early would be very good. So it's good to identify children young. It's also helpful for the family to know that they have a risk of having AT children, so they maybe don't have another child with AT. But the bad part of this is that, of course, an AT family now has their life changed very, very terribly much earlier. And today we don't have a treatment. So it's good and bad that we are diagnosing earlier. We spend a lot of effort, and Jennifer, who's on the phone, is wonderful at encouraging AT families to participate in AT research. This is important because sometimes companies or academic investigators have a potential therapeutic, and they want to pick a disease where it will be easy to do the trial and where recruiting will be easy. And so we like to be able to tell them, don't worry, we can definitely recruit patients to participate in your trial. We've interacted with many government agencies like the National Institutes of Health, NIH, and also the Food and Drug Administration, FDA. And we also learn from a lot of other nonprofit organizations. So in the United States, there's an organization called the Michael J. Fox Foundation, which is very large and funds research on Parkinson's disease. But they have been fantastic, very good at developing all kinds of research tools that are helpful for their disease. And so we learn from them and we watch and speak to them frequently. More recently, with digital technologies, we've also begun developing wearable sensors. So either a watch or a bracelet for the ankle, but technologies that can maybe improve the measurement of AT, particularly the neurological phenotype. So this is a slide I won't describe in great detail, but as you can see, we really believe that with enough detail, instead of having a child visit a neurologist one day when maybe it's a good day, maybe it's a bad day, we can have a child wear a digital device for weeks or months and collect all the data, and then use machine learning algorithms and other approaches, big data approaches, to try to extract patterns that maybe a typical human cannot detect. So that is our goal. So far, we have seen, several papers have been published, we have seen that a signature for AT has emerged, and we can use that to in future clinical trials. So now the part that is a little bit frustrating for all of us is that, the ATM protein plays a very central role in cellular function and sensing and coordinating the cell's response to DNA damage. It has many, many substrates, proteins it interacts with, or phosphorylates. Some are very well known in some areas like cancer. ATM acts on P53, BRCA1, the breast cancer gene, P53 mutated in most tumors. But this is fascinating and makes it really exciting for cell biologists. Many of you are probably very interested in this. But for a father like me, this is very frustrating. My wish is that it would be much more simple. It would be just one protein that below ATM that we need to either activate or inhibit. Over time, this has gotten even more complicated. This is still about 10 years ago. Today, using new technologies, with single cell sequencing and other kinds of massively parallel approaches, even this chart is even bigger. And you have to keep in mind that for most of these nodes on this chart represents a lot of work by somebody's lab who specializes in that protein. So tremendous amount of literature has been published. But the frustration is, out of all this biology, we need to identify, a path forward, a protein perhaps that's overexpressed, that if inhibited, it could normalize cells. That's a challenging strategy. There are a lot of questions we still have about AT and why brain cells are dying in AT. The general model has always been that DNA damage in neurons that are not dividing is accumulating, and that DNA damage reaches a threshold, causing the cells to eventually die. Oxidative stress, we now know that AT cells generate oxidative stress separate from the DNA damage response, and so could that be it? And there's an idea that some researchers have that even though they're supposed to be in G0, non-dividing cells, that neurons in AT are put back into cell cycle, causing them to die. We know that vesicle function is a problem in vesicle trafficking is a problem in AT cells. That could be a reason for neurons dying. I'll talk in a few minutes about neuroinflammation in AT. And even protein degradation, even though it's more common in some other neurodegenerative diseases, there's some evidence for it in AT. At this time, there are a few companies that are running clinical trials in AT. I'm sure you're aware of them. For years, there have been anecdotal evidence that low-dose steroids, AT children who were put on steroids for other reasons, their families said, well, that child has a little bit better improvement in energy or maybe in speech. And so there are several companies that are advancing programs for low-dose steroids. It's been a little bit frustrating for me. In the early days, there were some trials done in Italy where they were open label, not really placebo control. And then they did a placebo controlled trial, but it was very small, not very well powered. And so we're finally getting some answers, and it appears that low-dose steroids does have some efficacy, but the effect size is quite small. There's also some other approaches with nutraceuticals or metabolic approaches, and so N-acetyl-L-leucine by Intrabio is a company that's trying it in a basket trial on a lot of different diseases, and now they're trying an AT trial. And Ultragenyx has a compound called Dojolvi or triheptanoin. Martin Lavin, some of you know, in Australia, has been with David Coleman there running a trial. Has an interesting, unusual primary readout of epithelial cells in the nose that they use as the readout for primary endpoint, but they have many secondary endpoints, and some of those they think are showing some efficacy. I'm not sure. The paper has not been published. But for us right now, I think that the approach that seems to have the most excitement is addressing the actual cause of AT. So whether that's gene replacement or somehow editing the gene or silencing the gene seems to be the most exciting approach. So these categories would be, gene therapy using a vector like AAV or lentivirus, gene editing using CRISPR and other approaches. Today, we are funding someone in David Liu's lab at Harvard who's doing base editing and prime editing in hopes of being able to, for some subset of AT kids, at least maybe be able to correct their mutations. As all of you know, I think most of you know, there are 2 children with Turkish backgrounds, one in the US, one in Germany, who are now being treated with a ASO to silence a particular mutation. There are other approaches besides ASOs, such as transfer RNA approaches, which allow you to swap out a codon and basically skip over a mutation. And then there are less specific therapeutics like read-through compounds that have a problem because they're not specific, and toxicity is an issue, but could allow some families to maybe have their children skip a mutation. There's a lot that we don't know yet about AT gene that would be very helpful. For example, we don't really have strong data about whether every exon is necessary in every cell type in the body. So there are 66 exons in ATM. Do you really need exon 33? If you have a frame shift, a deletion and a frame shift in an exon that causes a truncation of the protein, and you knew you could live without that exon, it would be quite straightforward, quite easy to develop an ASO to skip that exon. So there are a lot of things we need to understand better. Right now, we're funding a researcher in Germany who is doing the experiments to systematically skip one exon at a time in AT, in the ATM, and looking at that in several types of cells, such as neurons derived from iPS cells. The biggest challenge, as you know, for ATM delivery is the large size of AT. ATM is about 150, 145 kb genomic size, and the coding sequence is about 9.2 kb. Today, the AAV vectors, the largest package they can deliver is about 4.3 kb, I believe. So it seems like it's difficult. Some people have published that in AT, you can maybe make a smaller gene that could still have some function, but we have not seen enough data to convince us of that. It seems like, particularly because most of the functional motifs are near the end of the gene, that you need the whole gene. One solution that some companies are pursuing right now is a multi-vector approach. So if you could take pieces of the ATM gene and several, 2 or 3 vectors and deliver it, and then put the gene back together, it could be maybe produce a functioning ATM protein. There are issues with that. You have to make sure that the fragments you create by themselves cannot be deleterious or toxic, but that's another strategy that we're exploring. Again, I won't go through this list, but there are many other challenges besides that for gene therapy, delivery into the brain, making sure you don't have immunogenicity, off-target effects, safety, and we've been talking a lot to the regulatory authorities, particularly with ASOs, where if the sequence is the only thing that's changed, but everything else is the same, do we have to do all of the toxicology studies to get approval for a drug, or could they make that easier or shorter? And long-term efficacy. A lot of gene therapy that you read about in literature, the problem is the durability is quite short, and the cost is high. So most of you know the story about Timothy Yu at Boston Children's Hospital. We're in a really short amount of time, about 18 months, went from knowing the mutation to developing an ASO and dosing the first child. We now have 2 children. Based on wearable devices and neurological exams, the little girl seems to be doing better. It's a little early to celebrate, but we're getting very excited. I think one of the reasons that Claudio visited Turkey a few weeks ago was to meet other AT children with the same mutations to see if the phenotype was in any way mild or if it was classic. It sounds like it was classic. Besides Boston Children's Hospital, there's a group in Australia that's developing an ASO for certain mutations. Mathis Snovik in Germany is also developing multiple ASOs for AT. Besides these efforts, another group at St. Jude Children's Hospital in Tennessee is now pursuing 3 or 4 ASOs for AT. But the thing to keep in mind is right now, based on our understanding of ASO technology and the mutation spectrum that is available, the catalog of mutations seen in AT children, even if we have the funding and the technology to develop an ASO for every mutation that is tractable with an ASO approach, we can still only address maybe 25% of AT children in the world. So even though we are very enthusiastic in supporting ASOs, we are also, of course, looking at other strategies, and ASOs are not enough. One of the ideas I had was that we could make a PET tracer against ATM, so that if you deliver an ASO, for example, you could have that, you could determine if that, ASO reached the brain, reached the cerebellum, if it in fact was taken up and transcribed, and if functioning protein was being made long before you could see a clinical benefit. So it'd be very, very helpful for clinical trials if you could more quickly at least determine if you have target engagement, if you have reached the right tissues and gotten expression. So right now, there's a company called Kudos that Steve Jackson in United Kingdom started. He sold it to AstraZeneca, a large drug company. They have an ATM inhibitor compound. So they had another compound that was also an AT inhibitor that has a very short half-life. So we persuaded them to allow a researcher in Boston to take that compound and radio label it. And at this point, right now, it's being tested in non-human primate monkey models to see if it is successful in labeling the ATM protein. If it is, the theory is that if you PET scan an AT child, it should be very dark. And if you PET scan an AT child who has received ASO therapy, hopefully you can see the ATM protein light up. So we have other approaches that we're taking besides the genetic approach to AT treatments. There's some non-specific things like anything you could do with growth factors to keep the brain cells from dying without addressing the actual cause. This is a challenge, of course, because you may not want to give growth factors to children with a disease that has a very high cancer risk, so it has to be very specific. Inflammatory response, there's clear evidence of neuroinflammation in AT and also mitochondrial dysfunction. So in mitochondria, AT cells we know produce harmful reactive oxygen species. They struggle to remove damage. Mitochondria, they struggle to maintain homeostasis, the balance between making and breaking apart mitochondria. You see that is disrupted in AT, and really biogenesis of mitochondria is limited. So we know there are mitochondrial problems in AT. We think that this could play a big role in the cerebellar degeneration because, for example, Purkinje cells are very large, cells with very high metabolic activity. Purkinje cells have a lot more mitochondria than most cells, and they require a constant energy because they're constantly firing. And they also have this complex branching structure that really requires mitochondria to move to certain places, particularly the synapses. So mitochondrial dysfunction could possibly explain the vulnerability of Purkinje cells, in AT. And then there are many approaches. I won't go through this list now, but there are many approaches that if you think it's a mitochondrial dysfunction that you could maybe try in AT, addressing the oxidative stress. There are a lot of catalytic approaches people have for that. Restoring energy production by increasing NAD. Many years ago, we funded a research lab that tried nicotinamide in AT cells and found that it increased NAD and helped cell survival. And so on this list, what we have to do is figure out how do we prioritize this? And if there are companies that are doing some of these approaches, can we persuade those companies to become interested in AT? Neuroinflammation is an area that we're particularly interested in now. As many of you know, in neurodegenerative diseases in general, there are increasing evidence of neuroinflammation playing a role, not just a side effect, but possibly exacerbating or making the disease worse. Regardless of the cause of your disease, if it's Alzheimer's, it's caused by genetics or caused idiopathically, or even plaques forming. If there's potentially a runaway or overreaction and inflammatory response, could that make the disease worse? And so in AT, we have clear evidence of elevated inflammatory cytokines. There's a long list of increasing evidence that inflammation is playing a role in AT. We would like to test this out, understand it better. There again because this approach is now interesting to many other neurodegenerative diseases, there are many drugs being developed, and it may be possible to try one of these on AT kids. Does not matter which mutation the child has, it would be good for all children with AT. And as I mentioned earlier, at this moment, we're trying to do a TSPO scan, PET imaging scan, to see if we can have something that's measurable in the brain revealing inflammation. I'm not so sure about that scan, but if it does work, then it would be a wonderful biomarker. And we're planning in the fall this year to have a workshop, just as we did last year in gene therapy, this year on neuroinflammation in AT, where maybe half the people will be experts on AT, and the other half will be people who are experts on neuroinflammation and therapeutics targeting neuroinflammation. And then I wanted to tell you the story of a company called Adrestia that was recently bought by another company called Insmed. So this is Steve Jackson, who's a longtime AT researcher at Cambridge University. He's always been interested in DNA PK and AT pathways. And he decided to use a CRISPR screen to systematically knock out every gene in the genome one at a time to try to identify genes that when knocked out would be therapeutic and would somehow normalize the phenotype of AT cells. And it depends on which things you're assaying, so he could only pick certain assays. But in the process of doing that, they identified two genes that instead of synthetic lethality, I think it's called synthetic rescue, but the two genes that when they knock those genes out, it makes AT cells more normal again. One one of the things they did was they not only did they test it in a lot of more biological assays, but they also looked in our database of AT children in our data platform. In those genes, not in the ATM gene, but in those two genes they found as potential targets, to see if there were any variants in those genes that were also associated with milder phenotype. And one of those genes did, in fact, have variants that were associated with milder phenotype. So it's very exciting. The company Insmed is now making a small molecule drug to inhibit that target protein. And it's a potential treatment for AT. This slide just reminds you that developing a new drug has many, many steps and a lot of high costs. We work with a lot of academics that get us to the early part of this to validate a target, but often you need companies to come in and get involved to handle the next steps, to all the clinical development, the toxicology, the CMC, control manufacturing, everything. But so this is discouraging. However, one idea we had was that maybe there are some existing drugs that have already completed those steps. Maybe they are already marketed and available to physicians, or maybe they were very late stage drugs that failed for some other disease. But those drugs have already had a lot of work done. So what we decided to do is, with a company called Charles River Labs, we ran a high throughput screen of about 5,000 compounds, which represent all approved compounds and compounds that have been in late stage development, to try to find compounds that could somehow improve the AT phenotype in cells. In this case, the assay we used was just activation of CHEK2, a downstream substrate of ATM. But this strategy could be used with a lot of other assays. We came up with eight hit compounds. And at this moment, we're having a lot of bioinformatics people try to understand better what the similarities are between these 8 compounds and what we can learn from them, and maybe to prioritize them. So still another idea, separate from the biologists of the world and the geneticists, is to recognize that maybe there's nothing we can do about the cell loss in AT. Maybe it's too late, and the Purkinje cells are, from early development, are already in trouble. But if the Purkinje cells are lost in the cerebellum, because of what we understand about the circuitry, maybe there's something else we can do that can improve movement control or delay it a little bit. And so, for example, in Purkinje cells in the cerebellum, you have, in a healthy cerebellum, the Purkinje cells inhibit cells in the deep cerebellar nuclei. If you have AT and you lose Purkinje cells, perhaps the deep cerebellar nuclei are hyperactive. And if you could confirm that, maybe you could pharmacologically, or with maybe an electronic approach like deep brain stimulation, somehow inhibit the hyperactivity of the deep cerebellar nuclei. So this idea of looking at the circuitry is important to us. There's also some new technologies coming along with old approaches like focused ultrasound and transcranial magnetic stimulation and deep brain stimulation that are continuing to make progress. And so we at this moment are close to starting a clinical trial for a focused ultrasound in AT. We know that a couple of AT kids in the United States have had deep brain stimulation because they had dystonia, but we're looking to see if that deep brain stimulation helped some maybe with the other symptoms of AT. And then for a really crazy idea, the most crazy idea I can tell you is separate from therapeutics. We're also wondering, is there ways that engineers could help improve the quality of life for AT children? So many of you have seen exoskeletons on the internet. These are usually designed for people who have paralysis or no muscle strength. In AT, we don't have a problem with muscle strength. We have a problem with control. But there are many engineers out there that are working with gyroscopes to balance things. There are people who are developing humanoid robots that you can push, and the robot begins to fall but stops falling and balances. So even for an AT child who is sitting and has trouble with truncal stability, perhaps there's a vest that can be made that can help stabilize using gyroscopes and accelerometers. And so one of our strategies at the AT Children's Project now is to also encourage engineers to look at our children's lives and determine, is there anything that can be done to improve quality of life? You know, in Parkinson's, they have a spoon that has like noise canceling technology. So the spoon is stable for soup even when you have tremor. Maybe there are similar approaches we can do with AT. So as I mentioned earlier, one of the things that the AT Children's Project does is also now encouraging biotechnology companies and pharmaceutical companies to become involved. Even though the AT children is a very small market, no company is interested in such a small market for a product. But many early stage companies actually want to test their drug on a disease where they can get a proof of concept very early and very quickly. So even though AT is rare, I believe that we can now begin to get drug companies and biotechnology companies who have a lot of funding to support research on AT. So we've been making a lot of efforts at the AT Children's Project. We had a big fancy ball last spring where we brought about 250 people together, all from the biotech and pharmaceutical industries. And for one night, all these executives and leaders of companies learned about AT. So meanwhile, I'm almost done, but I wanted to tell you that although I've been a volunteer and I do that my ninth job. In my day job, I also wanted to think about therapeutic development full time. I could not run my food business, and one phone call is about the food industry, and the next is about AT. It's very frustrating. And my heart and my passion was in trying to do something for AT. And so I decided to change my career. And so I switched from my food industry to starting a company in California called Perligen. That was the one of the first companies to pursue whole genome association studies on a very large scale using microarray technology. And then in 2009, I started a company with 3 neuroscientists from Rockefeller University called Envoy Therapeutics. So at the genomics company, I couldn't think about AT, but I could at least have PhDs and MDs work with me. I could ask them questions all the time. It was very helpful. Envoy Therapeutics was a company that had a unique technology with transgenic mice, but it focused on neurological diseases. So I could now have neuroscientists to learn from all the time. And most recently, I started a company called Ceravance, which has profiled, using RNA-seq and transcriptional and epigenetic technologies, over 16,000 postmortem human brain tissue samples. So a lot of brain tissue scanning and including AT. And I get to think about neuroscience all the time. So my career, I've kind of forced my career to be closer to AT, but AT is still what is most important to me. I've also been involved with some government and other company boards related to therapeutics development. But for me, I still think about AT when I fall asleep at night and when I wake up in the morning. And all of the things I described to you are still moving too slow, much too slow, and never fast enough. So that's why I'm excited that to have all of you on the phone today. I think it's in AT families across the United States are very scattered, and it sounds like in Turkey, you've got these pockets where some of you see a lot of AT patients. Sounds like you have first-rate science going on. So it seems like there's a great opportunity to do more things in Turkey for AT research, and I'm eager to help and collaborate any way we can. So last I wanted to say that I wish that I could visit you in person today, but maybe sometime soon we can visit. Thank you very much for letting me speak, and that's my son in the middle with 4 AT ladies. Thank you very much.